Track 02. Physics. Physics questions usually ask for one of four things: motion, force, energy, or waves and electricity. Speed equals distance divided by time. Use speed equals d over t when motion is scalar and direction does not matter. Velocity is displacement divided by time. Use velocity when direction matters. Acceleration equals change in velocity divided by time. Use acceleration equals final velocity minus initial velocity divided by time. For motion graphs, slope of position versus time gives velocity. Slope of velocity versus time gives acceleration. Area under a velocity, time graph gives displacement. Force equals mass times acceleration, F equals ma. Use it when a net force causes acceleration. Weight equals mass times gravitational field strength. W equals mg. Mass is matter. Weight is gravitational force. Newton's first law. No net force means no change in motion. Newton's second law. Net force equals mass times acceleration. Newton's third law. Forces come in equal and opposite pairs acting on different objects. Work equals force times distance in the direction of motion. W equals Fd. Use work when a force moves an object through distance. Power equals work divided by time. P equals W over T. Use power when the question asks how fast energy is transferred or work is done. Kinetic energy equals 1/2 mv squared. Use Ke equals 1/2 mv squared for energy of motion. Velocity is squared, so doubling speed quadruples kinetic energy. Gravitational potential energy equals mgh. Use Pe equals mgh for stored energy due to height. Mechanical energy is kinetic plus potential. If friction is ignored, mechanical energy is conserved. Momentum equals mass times velocity. P equals mv. Use momentum for collisions, recoil, impulse, and moving objects interacting. Impulse equals force times time and also equals change in momentum. Use impulse when force acts during a time interval. Thermal energy depends on particle motion and amount of matter. Temperature measures average kinetic energy. Heat is energy transferred because of temperature difference. Conduction is heat transfer by direct contact. Convection is heat transfer by fluid motion. Radiation is heat transfer by electromagnetic waves. Wave speed equals frequency times wavelength. V equals F lambda. Use this when frequency and wavelength appear. Frequency is cycles per second. A unit is hertz. Wavelength is distance between matching points on a wave. Amplitude relates to energy. Sound is a longitudinal mechanical wave. It requires a medium. Light is an electromagnetic transverse wave. It can travel through vacuum. For electromagnetic radiation, higher frequency means higher energy and shorter wavelength. Energy of a photon equals Planck's constant times frequency. E equals hf. Ohm's law is voltage equals current times resistance. V equals IR. Use it for circuits. Electric power equals voltage times current. P equals VI. Also, P equals I squared R, and P equals V squared over R when substitutions are needed. In a series circuit, current is the same everywhere. Total resistance increases by addition. Voltage is shared. In a parallel circuit, voltage is the same across each branch. Total resistance decreases when branches are added. Current splits. Gravity acts on mass and only attracts. Electric force acts on charge and can attract or repel. Like charges repel, opposite charges attract. Universal gravitation uses F equals gm1m2 divided by r squared. Coulomb's law uses F equals kq1 q2 divided by r squared. Both are inverse square laws. If distance doubles, force becomes 1/4. Quantum physics. Electrons occupy energy levels. When electrons absorb energy, they move to higher levels. When they fall to lower levels, they emit photons. Line spectra are evidence for quantized energy. Photoelectric effect means light can eject electrons from metal only if photon frequency is high enough. Increasing intensity below threshold frequency does not eject electrons. Track 03. Chemistry. Chemistry questions usually ask about particles, formulas, reactions, energy, or charge. Matter is anything with mass and volume. A pure substance has fixed composition. Elements contain one type of atom. Compounds contain chemically bonded elements. Mixtures are physically combined and variable in composition. Physical change changes form, phase, or appearance without changing identity. Chemical change produces a new substance. Density equals mass divided by volume. D equals m over v. Use density when mass and volume are linked. Atomic number equals protons. In a neutral atom, electrons equal protons. Mass number equals protons plus neutrons. Neutrons equal mass number minus atomic number. Isotopes have the same protons and different neutrons. Ions have gained or lost electrons. Cations are positive because electrons were lost. Anions are negative because electrons were gained. Average atomic mass is a weighted average of isotope masses based on abundance. Periodic trends. Atomic radius increases down a group and decreases left to right across a period. Ionization energy increases up and to the right. Electronegativity increases up and to the right, with fluorine highest. Metals lose electrons. Nonmetals gain or share electrons. Ionic bonds formed by electron transfer, usually metal to nonmetal. Covalent bonds formed by electron sharing, usually nonmetal to nonmetal. Metallic bonding involves metal cations in a sea of delocalized electrons. Lewis structures show valence electrons. The octet rule means many atoms tend toward 8 valence electrons. Polarity depends on electronegativity difference and molecular geometry. A polar bond can exist in a nonpolar molecule if the shape is symmetrical and dipoles cancel. Gas laws connect pressure, volume, temperature, and moles. Boyle's law, pressure and volume are inverse when temperature and moles are constant. P1 V1 equals P2 V2. Charles's law, volume and Kelvin temperature are direct when pressure and moles are constant. V1 over T1 equals V2 over T2. Gay-Lussac's law, pressure and Kelvin temperature are direct when volume and moles are constant. P1 over T1 equals P2 over T2. Combined gas law, P1 V1 over T1 equals P2 V2 over T2. Use Kelvin for all gas temperature calculations. Ideal gas law, PV equals nRT. Use it when pressure, volume, moles, and temperature appear together. Moles are the bridge in chemistry. Molar mass converts grams to moles. Avogadro's number converts particles to moles. Balanced equations convert moles of one substance to moles of another. Molarity equals moles of solute divided by liters of solution. M equals N over V. Use it for concentration. Dilution uses M1V1 equals M2V2. Moles of solute stay constant while volume changes. Stoichiometry path, grams to moles, mole ratio, moles to requested unit. Limiting reactant is the reactant that runs out first. Percent yield equals actual yield divided by theoretical yield times 100%. Reaction rate increases with higher temperature, higher concentration, greater surface area, and catalysts. A catalyst lowers activation energy and is not consumed. Equilibrium means forward and reverse reaction rates are equal. Le Chatelier's principle. A system at equilibrium shifts to reduce stress. Acids increase hydronium or donate protons. Bases increase hydroxide or accept protons. pH equals negative log of hydrogen ion concentration. Lower pH means more acidic. Redox means electron transfer. Oxidation is loss of electrons. Reduction is gain of electrons. Oil rig. Oxidation is loss. Reduction is gain. Oxidation number increases during oxidation. Oxidation number decreases during reduction. Nuclear chemistry changes the nucleus. Alpha decay emits a helium nucleus. Beta decay changes neutron-proton balance. Gamma emission releases energy. Half-life is the time for half of a radioactive sample to decay. Track 4. Biology questions usually ask how structure supports function, how information is copied and expressed, how energy moves, or how systems maintain homeostasis. Water matters because it is polar. Polarity gives water cohesion, adhesion, high specific heat, and solventability. Carbohydrates provide quick energy and structural support. Lipids store long-term energy and form membranes. Proteins provide structure, transport, signaling, movement, and enzymes. Nucleic acids store and transmit genetic information. Enzymes are proteins that lower activation energy. Shape determines function. Active site shape determines substrate fit. Denaturation changes shape and reduces function. Cells are the basic unit of life. Prokaryotes lack a nucleus and membrane-bound organelles. Eukaryotes have a nucleus and membrane-bound organelles. Nucleus stores DNA. Ribosomes build proteins. Rough endoplasmic reticulum modifies proteins. Smooth endoplasmic reticulum makes lipids and detoxifies. Golgi apparatus sorts and packages. Mitochondria perform cellular respiration. Chloroplasts perform photosynthesis. Cell membrane regulates entry and exit. Cell wall provides support in plants, fungi, and many prokaryotes. Passive transport moves down a concentration gradient and does not require cellular energy. Diffusion moves particles from high to low concentration. Osmosis is diffusion of water across a membrane. Facilitated diffusion uses transport proteins but still moves down the gradient. Active transport moves against a concentration gradient and requires energy. Photosynthesis stores energy. Carbon dioxide plus water plus light produces glucose plus oxygen. It occurs in chloroplasts. Cellular respiration releases usable energy from glucose. Glucose plus oxygen produces carbon dioxide, water, and ATP. It occurs mainly in mitochondria. Aerobic respiration uses oxygen and produces more ATP. Anaerobic respiration or fermentation occurs without oxygen and produces less ATP. Cell cycle includes growth, DNA replication, and division. Mitosis makes genetically identical body cells. Meiosis makes gametes with half the chromosome number and increases genetic variation. DNA stores genetic code. RNA helps express it. Transcription makes RNA from DNA. Translation makes protein from RNA at the ribosome. Codons specify amino acids. Amino acid sequence determines protein folding. Folding determines active site or functional shape. Shape determines function. Genotype is allele combination. Phenotype is observable trait. Dominant alleles mask recessive alleles in simple inheritance. Recessive traits appear when two recessive alleles are present. Incomplete dominance blends phenotype. Codominance shows both alleles. Sex-linked traits are often on the X chromosome and show different inheritance patterns in males and females. Mutations are DNA changes. Some are harmful, some neutral, some beneficial. Mutation creates variation. Natural selection acts on variation. Evolution is change in populations over generations. Individuals do not evolve. Populations evolve. Natural selection requires variation, heritability, overproduction, competition, and differential survival or reproduction. Classification organizes life by shared traits and evolutionary relationships. 3 domains are bacteria, archaea, and eukarya. Homeostasis means regulated stability, not no change. Negative feedback reverses a change. Positive feedback amplifies a change until an endpoint. Structure-function rule. Form explains function. In every anatomy or physiology question, ask what structure is present and what job it makes possible. Track 5. Ecology, Earth systems, and space. Ecology questions ask how matter cycles, how energy flows, and how populations change. Energy flows one way through ecosystems. Matter cycles. Producers convert sunlight or chemical energy into organic molecules. Consumers get energy by eating other organisms. Decomposers recycle matter. Food chains show one path of energy. Food webs show many feeding relationships. Only a fraction of energy transfers between trophic levels. Energy pyramids narrow at higher levels because energy is lost as heat and life processes. Population growth depends on birth rate, death rate, immigration, and emigration. Carrying capacity is the maximum population an environment can support long term. Limiting factors restrict population size. Density-dependent factors include disease, competition, predation, and parasitism. Density-independent factors include drought, flood, fire, and temperature extremes. Symbiosis means close interaction. Mutualism benefits both. Commensalism benefits one without significantly affecting the other. Parasitism benefits one and harms the other. Carbon cycles through photosynthesis, respiration, decomposition, fossil fuels, combustion, oceans, and atmosphere. Nitrogen cycles through fixation, nitrification, assimilation, ammonification, and denitrification. Biodiversity increases ecosystem stability because more species can fill roles and buffer disturbances. Earth systems interact. Geosphere, hydrosphere, atmosphere, and biosphere exchange matter and energy. The rock cycle transforms rock through melting, cooling, weathering, erosion, deposition, compaction, cementation, heat, and pressure. Igneous rock forms from cooled magma or lava. Sedimentary rock forms from compacted and cemented sediments. Metamorphic rock forms when existing rock changes under heat and pressure without melting. Weathering breaks rock down. Erosion moves material. Deposition drops material. Plate tectonics explains earthquakes, volcanoes, mountain building, ocean basins, and continental movement. Divergent boundaries move apart and often create new crust. Convergent boundaries move together and can form mountains, trenches, volcanoes, or subduction zones. Transform boundaries slide past and often produce earthquakes. Relative dating orders events. Absolute dating estimates age, often using radioactive decay. Index fossils are widespread, abundant, and limited to a short geologic time span. Weather is short-term atmospheric condition. Climate is long-term pattern. Convection in the atmosphere and oceans moves heat. Uneven solar heating drives wind, ocean circulation, and weather patterns. Space questions ask about motion, gravity, light, and scale. Rotation causes day and night. Revolution around the sun helps define the year. Seasons are caused by Earth's axial tilt, not changing distance from the sun. Moon phases are caused by the changing visible portion of the moon's sunlit half. Eclipses require alignment. Solar eclipse, moon between Earth and sun. Lunar eclipse, Earth between sun and moon. Tides are mainly caused by gravitational interaction with the moon, with the sun also contributing. Stars form from nebulae. Main sequence stars fuse hydrogen into helium. Massive stars can become supernovae. neutron stars, or black holes. Lower mass stars can become red giants, planetary nebulae, and white dwarfs. Hotter stars are bluer. Cooler stars are redder. Brightness depends on temperature, size, and distance. The Big Bang model explains an expanding universe from an early, hot, dense state. Evidence includes cosmic microwave background radiation, redshift of galaxies, and abundance of light elements. Track 6. Instruction, assessment, and equation identification. Science instruction questions ask what a teacher should do to reveal student thinking and improve learning. Use inquiry when students need to investigate a question using evidence. Use direct instruction when safety, procedure, or a new technical model must be clear first. A misconception is not a lack of knowledge. It is an incorrect mental model. Good assessment reveals the model, not just the answer. Formative assessment happens during learning and guides instruction. Summative assessment evaluates learning after instruction. A good science assessment asks students to use evidence, models, data, or reasoning. Memorization alone is weak evidence of understanding. CER means claim, evidence, reasoning. Claim answers the question. Evidence uses data or observations. Reasoning explains why the evidence supports the claim using science principles. For accommodations, keep the science target and reduce unnecessary barriers. Use word banks, sentence stems, chunked directions, visuals, reduced copying, and extended time when appropriate. Equation identification is a pattern skill. If the problem gives mass and volume, think density equals mass divided by volume. If the problem gives distance and time, think speed equals distance divided by time. If the problem gives initial velocity, final velocity, and time, think acceleration equals change in velocity divided by time. If the problem gives mass and acceleration, think force equals mass times acceleration. If the problem gives force and distance in the direction of motion, think work equals force times distance. If the problem gives work and time, think power equals work divided by time. If the problem gives mass and speed, think kinetic energy equals 1/2 mass times velocity squared. If the problem gives mass, gravity, and height, think gravitational potential energy equals mass times gravitational field strength times height. If the problem gives frequency and wavelength, think wave speed equals frequency times wavelength. If the problem gives voltage, current, and resistance, think Ohm's law, voltage equals current times resistance. If the problem gives pressure, volume, and temperature for the same gas before and after change, think combined gas law. If the problem gives pressure, volume, moles, and temperature, think ideal gas law. If the problem gives moles of solute and liters of solution, think molarity equals moles divided by liters. If the problem changes concentration by adding solvent, think dilution. M1 V1 equals M2V2. If the problem asks grams to grams in a reaction, think stoichiometry. Grams to moles, mole ratio, moles to grams. If the problem asks actual yield compared to theoretical yield, think percent yield equals actual divided by theoretical times 100%. If the problem asks radioactive decay over time, think half-life. Remaining amount equals original amount times 1/2 raised to the number of half-lives. If the problem asks photon energy, think energy equals Planck's constant times frequency. If the problem asks gravity or electric force and distance changes, think inverse square. Double distance means 1/4 force. Triple distance means 1/9 force. Final rule: identify the quantity requested first Then identify the variables given, then choose the equation whose variables connect the given information to the requested quantity. Track 07, a chemistry. Math map, what equation do I need? Chemistry math is not random. Every problem gives a path. Your job is to identify the quantity asked for, identify the quantities given, then choose the equation that connects them. Start with the picture. If the problem gives mass and volume, picture a box filled with matter. The question is how tightly packed the matter is. Use density. Density equals mass divided by volume. D equals m over v. Units usually look like grams per milliliter, grams per cubic centimeter, or kilograms per liter. If mass is missing, rearrange. Mass equals density times volume. If volume is missing, rearrange. Volume equals mass divided by density. The visual triangle is this: mass on top, density and volume on the bottom. Cover the missing variable. If mass is missing, density times volume. If density is missing, mass over volume. If volume is missing, mass over density. Next, atomic math. Atomic number equals protons. For a neutral atom, electrons equal protons. Mass number equals protons plus neutrons. Neutrons equal mass number minus atomic number. Picture the atom as two zones. The nucleus contains protons and neutrons. The electron cloud contains electrons. Mass number counts the heavy particles in the nucleus only. Protons plus neutrons. It does not count electrons. If the question gives atomic number 12, immediately say protons are 12. If the atom is neutral, electrons are 12. If the mass number is 25, neutrons are 25 minus 12, which is 13. Do not confuse atomic mass with mass number. Atomic mass on the periodic table is an average decimal. Mass number in a problem is usually a whole number for one isotope. Next, isotope average atomic mass. Average atomic mass equals isotope mass times fractional abundance plus isotope mass times fractional abundance, and so on. The key visual is a weighted average. Heavier or more abundant isotopes pull the average toward themselves. Convert percent abundance to a decimal before multiplying. 20% becomes 0.20, 80% becomes 0.80. If chlorine has about 75% chlorine 35 and 25% chlorine 37, the average must be closer to 35 than 37 because chlorine 35 is more abundant. Next, percent error. Percent error equals absolute value of measured minus accepted, divided by accepted times 100%. Use it when a lab measurement is compared to the true or accepted value. Picture the accepted value as the target. The measured value is where the dart lands. The difference is the miss. Divide by the target to scale the error. Next, percent composition. Percent composition equals part, divided by whole times 100%. For a compound, the part is the mass from one element. The whole is the molar mass of the entire compound. Picture a formula as a stack of element masses. Add all pieces for the whole. Then choose one element's piece and divide by the whole. For water, H2O, hydrogen contributes about 2 grams per mole, and oxygen contributes about 16 grams per mole. Whole is about 18 grams per mole. Percent oxygen is 16 divided by 18 times 100%. The equation map is simple. Packed matter, density, atom particles, protons, neutrons, electrons. Average isotope mass, weighted average. Lab accuracy, percent error, composition of compound, part over whole times 100. First, identify the picture, then choose the equation. Track 07, B, moles, molar mass, particles, and stoichiometry. The mole is the central bridge in chemistry. Picture a train station. Every chemistry math route passes through the mole station. Grams go to moles using molar mass. Particles go to moles using Avogadro's number. Liters of gas at STP go to moles using 22.4 liters per mole. Balanced equations connect moles of one substance to moles of another substance. Molar mass comes from the periodic table. Units are grams per mole. To convert grams to moles, divide by molar mass. Moles equals grams divided by molar mass. To convert moles to grams, multiply by molar mass. Grams equals moles times molar mass. Visualize the molar mass triangle. Grams on top, moles and molar mass on the bottom. Cover what is missing. Particles use Avogadro's number. 1 mole equals 6.022 times 10 to the 23rd particles. To convert particles to moles, divide by Avogadro's number. To convert moles to particles, multiply by Avogadro's number. For gases at STP, 1 mole equals 22.4 liters. To convert liters at STP to moles, divide by 22.4. To convert moles to liters at STP, multiply by 22.4. Now, stoichiometry. Stoichiometry is a balanced equation ratio problem. The coefficients in the balanced equation are mole ratios only. They are not gram ratios. The standard stoichiometry path is given unit to moles, mole ratio, moles to requested unit. If the problem starts with grams and asks for grams, the path is grams to moles, mole ratio, moles to grams. If the problem starts with particles and asks for grams, the path is particles to moles, mole ratio, moles to grams. If the problem starts with liters of gas at STP and asks for moles of another substance, the path is liters to moles, mole ratio. Picture the balanced equation as a recipe. Coefficients tell how many moles react or form. For example, 2H2 plus O, 2 produces 2H2O. The ratio of hydrogen to water is 2 to 2, or 1 to 1. The ratio of oxygen to water is 1 to 2. If the question gives moles of oxygen and asks for moles of water, multiply by 2 moles water over 1 mole oxygen. The unit you want goes on top. The unit you cancel goes on bottom. Limiting reactant problems ask which reactant runs out first. Picture 2 ingredients in a recipe. If you have too much of one and not enough of the other, the smaller useful amount limits the product. To find limiting reactant, calculate how much product each reactant can make. The reactant that makes less product is limiting. Theoretical yield is the maximum product predicted by stoichiometry. Actual yield is what was collected in the lab. Percent yield equals actual yield divided by theoretical yield times 100%. Visualize percent yield as real product over perfect product. Actual goes on top, theoretical goes on bottom. If actual is greater than theoretical, something is wrong. Wet product, contamination, measurement error, or calculation error. Core rule: when reactions are involved, convert to moles before using the balanced equation. Track 07C, gas laws, molarity, and dilution. Gas law problems are about pressure, volume, temperature, and moles. Always convert Celsius to Kelvin. Kelvin equals Celsius plus 273. Do not use Celsius in gas law calculations. Picture gas particles in a container. Pressure comes from collisions with the container walls. Temperature measures average kinetic energy. Volume is the space available. Moles are the number of gas particles. Boyle's law connects pressure and volume. P1 V1 equals P2 V2. Use Boyle's law when temperature and moles stay constant. Picture a syringe. Push the plunger in, volume decreases, particles hit the walls more often. Pressure increases. Pressure and volume move opposite directions. Charles's law connects volume and Kelvin temperature. V1 over T1 equals V2 over T2. Use Charles's law when pressure and moles stay constant. Picture a balloon. Heat it. Particles move faster and spread out. Volume increases. Cool it. Volume decreases. Volume and Kelvin temperature move the same direction. Gay-Lussac's law connects pressure and Kelvin temperature. P1 over T1 equals P2 over T2. Use Gay-Lussac's law when volume and moles stay constant. Picture a sealed rigid can. Heat it. Particles move faster, but volume cannot expand. Pressure increases. Pressure and Kelvin temperature move the same direction. Combined gas law connects pressure, volume, and temperature. P1 V1 over T1 equals P2V 2 over T2. Use it when pressure, volume, and temperature change, but moles stay constant. Ideal gas law connects pressure, volume, moles, and temperature. PV equals nRT. Use ideal gas law when the problem gives or asks about moles of gas and gives pressure, volume, and temperature. R depends on units. Commonly, R equals 0.0821 liter atmospheres per mole. Kelvin when pressure is in atmospheres and volume is in liters. Equation identification: pressure and volume only, Boyle. Volume and temperature only, Charles. Pressure and temperature only, Gay-Lussac. Pressure, volume, and temperature before and after. Combined gas law. Pressure, volume, moles, and temperature in one condition. Ideal gas law. Now, solutions. Molarity equals moles of solute divided by liters of solution. M equals N over V. Use molarity when concentration is measured in moles per liter. Picture solute particles spread through a liquid volume. More particles means higher molarity. More volume with the same particles means lower molarity. If moles are missing, moles equals molarity times volume. If volume is missing, volume equals moles divided by molarity. Volume must be in liters. Milliliters to liters, divide by 1,000. Dilution uses M1V1 equals M2V2. Use dilution when water is added and the amount of solute stays the same. Picture the same number of solute particles in a larger container. The particles are more spread out. Molarity decreases. In dilution, concentrated stock solution becomes less concentrated. M1 is usually larger than M2. V2 is final total volume, not the amount of water added. If the problem gives water added, then final volume equals original volume plus added water. Core rule: gas laws require Kelvin. Molarity requires liters. Dilution keeps moles of solute constant. Track 07D. pH, heat, energy, half-life, and redox math, acid-base math starts with pH. pH equals negative log of hydrogen ion concentration. pH equals negative log bracket H+. Low pH means high hydrogen ion concentration and stronger acidity. Each pH step is a factor of 10. A solution with pH 3 has 10 times more hydrogen ion than pH 4. It has 100 times more hydrogen ion than pH 5. pOH equals negative log of hydroxide ion concentration. At 25 degrees Celsius, pH plus pOH equals 14. If pH is 3, pOH is 11. Strong acid means high H+, strong base means high OH-. Neutral at 25 degrees Celsius is pH 7. Now heat. Specific heat problems use Q equals mc delta T. Q is heat energy, M is mass, C is specific heat. Delta T is final temperature minus initial temperature. Picture heat entering a substance. The temperature change depends on mass and specific heat. Water has high specific heat, so it takes more energy to change water's temperature. If Q is positive, heat is absorbed. If Q is negative, heat is released. Delta T equals final minus initial. A temperature increase gives positive delta T. A temperature decrease gives negative delta T. For phase changes, temperature stays constant while energy changes the state. Use QU equals MH fusion for melting or freezing. Use Q equals MH vaporization for boiling or condensation. If temperature changes, use Q equals MC delta T. If phase changes at constant temperature, use Q equals MH. Thermochemistry connects energy diagrams to sign. Delta H equals products minus reactants. If products are lower energy than reactants, delta H is negative. The reaction is exothermic. Heat is released. If products are higher energy than reactants, delta H is positive. The reaction is endothermic. Heat is absorbed. Activation energy is the energy barrier from reactants to the transition state. A catalyst lowers activation energy. It does not change delta H. It does not change the energy of reactants or products. It provides a lower energy pathway. Gibbs free energy predicts spontaneity. Delta G equals delta H minus T delta S. If delta G is negative, the process is spontaneous under those conditions. If delta G is positive, the process is non-spontaneous under those conditions. If delta G is 0, the system is at equilibrium. Now half-life. Remaining amount equals original amount times 1/2 raised to the number of half-lives. N equals N, 0 times 1/2 to the NN. N equals elapsed time divided by half-life. Picture cutting the sample in half again and again. After 1 half-life, 1/2 remains. After 2 half-lives, 1/4 remains. After 3 half-lives, 1/8 remains. After 4 half-lives, 1/16 remains. Do not subtract the same amount each time. Half-life is repeated halving, not repeated subtraction. Now redox. Oxidation is loss of electrons. Reduction is gain of electrons. Oxidation number increases during oxidation. Oxidation number decreases during reduction. Picture electrons moving from one substance to another. The substance that loses electrons is oxidized. The substance that gains electrons is reduced. Oil rig. Oxidation is loss, reduction is gain. If an element goes from 0 to +2, its oxidation number increased. It was oxidized. If an element goes from +5 to +2, its oxidation number decreased. It was reduced. For ionic compounds, total charge must balance. Group 1 metals are +1. Group 2 metals are +2. Aluminum is +3. Oxygen is usually -2. Halogens are usually -1. For formulas, crisscross charge magnitudes, then reduce to the lowest whole number ratio. Magnesium ion is +2. Chloride ion is -1. The neutral formula is MgCl2. Aluminum ion is +3. Oxide ion is -2. The neutral formula is Al2O3. Core rule. Chemistry math is unit logic plus particle logic. Follow the units, follow the particles, follow the charge. Track 07, E. Rapid equation identification drill density. Use when mass and volume are connected. D equals M over V. Percent error. Use when measured value is compared to accepted value. Percent composition. Use when element mass is compared to compound molar mass. Atomic number gives protons. Neutral atom, electrons equal protons. Mass number, protons plus neutrons. Neutrons, mass number minus atomic number. Average atomic mass, isotope mass, times fractional abundance added for all isotopes. Moles from grams, grams divided by molar mass. Grams from moles, moles times molar mass. Moles from particles, particles divided by Avogadro's number. Particles from moles, moles times Avogadro's number. Moles from gas volume at STP, liters divided by 22.4. Gas volume at STP from moles, moles times 22.4. Stoichiometry. Convert given to moles. Use mole ratio. Convert to requested unit. Percent yield, actual yield divided by theoretical yield times 100%. Boyle's law, pressure and volume change, temperature constant. P1 V1 equals P2 V2. Charles's law, volume and Kelvin temperature change, pressure constant. V1 over T1 equals V2 over T2. Gay-Lussac's law, pressure and Kelvin temperature change, volume constant. P1 over T1 equals P2 over T2. Combined gas law, pressure, volume, and temperature change. P1V1 over T1 equals P2V2 over T2. Ideal gas law, pressure, volume, moles, and temperature. PV equals nRT. Molarity, moles per liter. M equals N over V. Dilution, concentration, and volume before and after adding solvent. M1, V1 equals M2, V2. pH, negative log of hydrogen ion concentration. POH, negative log of hydroxide ion concentration. pH plus POH equals 14 at 25 degrees Celsius. Specific heat, Q equals MC delta T. Temperature change, delta T equals final temperature minus initial temperature. Phase change, Q equals MH fusion or Q equals MH vaporization. Enthalpy, delta H equals products minus reactants. Gibbs free energy, delta G equals delta H minus T delta S. Photon energy, E equals HF. Half-life, remaining equals original times 1/2 to the number of half-lives. Redox, oxidation is loss of electrons. Reduction is gain of electrons. Oxidation number increases in oxidation. Oxidation number decreases in reduction. Ionic formula, total positive charge plus total negative charge equals 0. Final procedure for every math question. Name the unknown. List the given values, identify units, choose the equation, rearrange before substituting, substitute with units, cancel units, calculate, check if the answer is reasonable. Reasonable answers matter. Density should match the scale of the material. Kelvin cannot be negative. Percent yield should usually be below 100%. Neutral ionic formulas must have 0 total charge. A gas heated at constant pressure should expand. A diluted solution should have lower molarity. A half-life sample should decrease by fractions, not by equal subtraction. Track 08A, cell structure and processes. Elements of life. Life is built from a small set of chemical elements arranged in precise molecular structures. The major elements of life are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Use the memory hook. CHONPS. Carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur. These elements matter because their atoms form the backbone, energy systems, information systems, and functional machinery of cells. Carbon is the central framework element. Picture carbon as a 4-way connector. Carbon has 4 valence electrons, so it can form 4 covalent bonds. This allows carbon atoms to form chains, rings, branches, and complex 3-dimensional structures. Hydrogen often fills open bonding positions. It helps shape organic molecules and affects acidity, polarity, and energy transfer. Oxygen is highly electronegative. Picture oxygen as pulling shared electrons toward itself. This creates polar bonds, helps form water, and appears in carbohydrates, lipids, proteins, and nucleic acids. Nitrogen is essential in amino acids and nucleic acids. Picture nitrogen as part of the language system of life. It appears in amino groups of proteins and nitrogenous bases of DNA and RNA. Phosphorus is essential in ATP, DNA, RNA, and phospholipids. Picture phosphorus as part of the cell's energy currency and membrane architecture. In ATP, phosphate groups store transferable energy. In DNA and RNA, phosphate forms the sugar phosphate backbone. In membranes, phosphate heads face water. Sulfur is found in some amino acids, especially cysteine and methionine. Sulfur helps stabilize protein shape. Picture sulfur atoms forming bridges that hold a folded protein in its working form. These elements combine through chemical bonds. Covalent bonds share electrons. They form the backbone of biological molecules. Ionic attractions occur between charged particles or charged regions. Hydrogen bonds are weak attractions involving hydrogen and electronegative atoms such as oxygen or nitrogen. Individually weak, collectively powerful. Van der Waals forces are very weak attractions between temporary charge differences. In large molecules, many small forces help stabilize shape. A biological molecule's function depends on its atomic arrangement, bond type, polarity, charge, and three-dimensional shape. The cell is not a bag of random chemicals. It is a structured chemical system. Elements form atoms. Atoms form molecules. Molecules form biomolecules. Biomolecules form cell structures. Cell structures perform life processes. When the exam asks about elements necessary for life, do not only name the elements, connect them to their biological jobs. Carbon builds skeletons. Hydrogen and oxygen help form water and organic compounds. Nitrogen builds proteins and nucleic acids. Phosphorus builds ATP, DNA, RNA, and phospholipids. Sulfur helps stabilize proteins. Life depends on chemistry because cells are molecular machines. Track 08B, water, carbon, and life processes. Water is essential because it is polar. Picture a water molecule as a bent V shape. Oxygen sits at the point of the V, two hydrogens angle outward. Oxygen pulls electrons more strongly than hydrogen. This gives oxygen a partial negative charge and the hydrogens partial positive charges. Because water is polar, water molecules attract each other. The slightly positive hydrogen of one water molecule is attracted to the slightly negative oxygen of another. This attraction is a hydrogen bond. Hydrogen bonds give water its life-supporting properties. Cohesion means water molecules stick to each other. Picture water molecules holding hands. Cohesion creates surface tension and helps water move as a continuous column through plant xylem. Adhesion means water sticks to other polar or charged surfaces. Picture water climbing along the inside wall of a narrow tube. Adhesion helps capillary action. High specific heat means water resists temperature change. Picture water as a thermal buffer. It absorbs a lot of heat before its temperature rises. This stabilizes body temperature, aquatic environments, and cellular conditions. High heat of vaporization means water requires significant energy to evaporate. When sweat evaporates, it removes heat from the body. This supports cooling. Water is less dense as a solid than as a liquid. Ice floats. This insulates bodies of water and protects aquatic life below. Water is an excellent solvent for polar and ionic substances. Picture water molecules surrounding ions. The oxygen end points toward positive ions. The hydrogen ends point toward negative ions. This separates and dissolves charged particles. In cells, water is the reaction medium. Most biochemical reactions occur in aqueous solution. Hydrophilic means water-loving. Hydrophilic substances are polar or charged and interact with water. Hydrophobic means water-fearing. Hydrophobic substances are nonpolar and avoid water. This hydrophilic hydrophobic difference organizes cells. Phospholipids form membranes because their phosphate heads are hydrophilic and their fatty acid tails are hydrophobic. Now, carbon. Carbon is significant because it forms 4 covalent bonds. It can bond to itself and to hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Picture carbon as molecular scaffolding. It can build straight chains, branched chains, rings, and complex folded structures. Carbon skeletons can vary in length, branching, double bonds, and attached functional groups. Functional groups change molecular behavior. Hydroxyl groups increase polarity. Carboxyl groups can act as acids. Amino groups can act as bases. Phosphate groups store and transfer energy and add negative charge. The properties of carbon allow diversity. The properties of water allow transport, reaction, temperature stability, and membrane organization. Carbon builds the structure. Water creates the environment. Together, water and carbon make life chemistry possible. Track 08C. Shape determines biological function. In biology, shape is not decoration. Shape is function. A molecule works because its shape allows specific interactions. Picture a key fitting into a lock. The key does not work because it is metal. It works because its shape matches the lock. Biological molecules work the same way. Enzymes have active sites. The active site is a specific 3-dimensional region where the substrate binds. The substrate is the reactant the enzyme acts on. If the substrate fits the active site, the enzyme can stress bonds, position atoms, and lower activation energy. If the active site changes shape, the enzyme may stop working. Temperature and pH can denature enzymes. Denaturation means the protein unfolds or changes shape. Picture a carefully folded paper crane being crumpled. The paper is still present, but the structure no longer performs the same function. DNA function depends on shape. DNA is a double helix. Picture a twisted ladder. The sugar phosphate backbones form the sides. Nitrogenous base pairs form the rungs. A pairs with TC pairs with G. This complementary base pairing allows DNA to copy itself. Each strand can serve as a template for a new strand. RNA shape also matters. tRNA has a folded shape that carries a specific amino acid and matches a codon during translation. Picture a tRNA as a molecular adapter. One part recognizes the RNA message, another part carries the correct amino acid. Receptors depend on shape. A receptor is a protein that binds a specific signal molecule. Picture a docking station on a cell membrane. A hormone or neurotransmitter fits into the receptor, triggering a response inside the cell. Neurotransmitters depend on shape. A neurotransmitter must fit the receptor at a synapse. If the shape or charge pattern matches, the signal continues. If not, the receptor does not activate. Lipids depend on shape and polarity. Phospholipids have a hydrophilic head and hydrophobic tails. In water, they self-assemble into bilayers. Picture a crowd of tadpole-shaped molecules. Heads face water, tails hide inside. This creates a membrane. Protein shape has levels. Primary structure is amino acid sequence. Secondary structure includes alpha helices and beta sheets. Tertiary structure is the overall 3-dimensional folding of one polypeptide. Quaternary structure is the arrangement of multiple polypeptide subunits. The amino acid sequence determines folding. Folding determines shape. Shape determines function. Hemoglobin carries oxygen because its folded protein shape creates binding sites for oxygen. Antibodies bind antigens because their variable regions have shapes that match specific targets. Channels and transport proteins move substances across membranes. because their shapes create selective passageways. The exam may ask why a mutation changes protein function. The answer is sequence affects folding. Folding affects shape. Shape affects function. The exam may ask why heat affects enzymes. The answer is heat disrupts bonds that maintain protein shape. A changed shape changes the active site. A changed active site changes function. Core rule: biological molecules perform jobs because their shapes and chemical properties match their targets. Track 08. D. Synthesis and degradation of biomolecules. Cells constantly build and break molecules. Synthesis means building larger molecules from smaller units. Degradation means breaking larger molecules into smaller units. Picture biomolecules as chains made of repeating building blocks. Monomers are small building blocks. Polymers are large molecules made of linked monomers. Carbohydrates are built from monosaccharides. Proteins are built from amino acids. Nucleic acids are built from nucleotides. Lipids are not true polymers in the same repeated chain sense, but many are assembled from glycerol and fatty acids. The key synthesis reaction is dehydration synthesis, also called condensation. In dehydration synthesis, two smaller molecules join and water is removed. Picture two train cars connecting. One loses an H, the other loses an O, H. H plus OH becomes water. The remaining parts bond together. Dehydration synthesis builds carbohydrates, proteins, nucleic acids, and many lipid structures. The key degradation reaction is hydrolysis. Hydrolysis uses water to break a bond. Hydro means water. Lysis means splitting. Picture water as molecular scissors. Water is added across a bond. One side receives H. The other side receives OH. The larger molecule splits into smaller pieces. Digestion is hydrolysis. When starch is broken into glucose units, hydrolysis occurs. When proteins are broken into amino acids, hydrolysis occurs. When nucleic acids are broken into nucleotides, hydrolysis occurs. When triglycerides are broken into glycerol and fatty acids, hydrolysis occurs. Cells use synthesis to build structures, enzymes, membranes, DNA, RNA, and storage molecules. Cells use degradation to digest food, recycle molecules, release energy, and remove damaged components. Chemical reactions require energy changes. Some reactions release energy, some require energy input. Activation energy is the energy needed to start a reaction. Enzymes lower activation energy, making reactions occur faster at cellular temperatures. Without enzymes, many life reactions would be too slow to sustain life. Biomolecule reactions are not just classroom formulas. They are life processes. DNA synthesis builds genetic material before cell division. Protein synthesis builds enzymes, transporters, receptors, antibodies, and structural proteins. Glycogen synthesis stores glucose in animals. Starch synthesis stores glucose in plants. Lipid synthesis builds cell membranes and long-term energy stores. Hydrolysis of food molecules provides absorbable units. Hydrolysis of ATP releases energy for cellular work. A TP breakdown is a hydrolysis reaction. ATP plus water becomes ADP plus inorganic phosphate plus energy. Picture ATP as a charged battery with 3 phosphate groups. Removing one phosphate releases energy that can drive cellular processes. Core rule: synthesis usually removes water to build. Hydrolysis uses water to break. Track 08E: biomolecules, structure, and function. There are 4 major biomolecule groups: carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates are made mainly of carbon, hydrogen, and oxygen. Their common ratio is approximately 1 carbon to 2 hydrogens to 1 oxygen. Carbohydrates function in quick energy, energy storage, and structural support. Monosaccharides are simple sugars, such as glucose. Disaccharides are two sugar units. Polysaccharides are long carbohydrate chains. Starch stores glucose in plants. Glycogen stores glucose in animals. Cellulose provides structural support in plant cell walls. Chitin provides structural support in fungal cell walls and arthropod exoskeletons. Picture carbohydrates as fuel blocks or support beams depending on their arrangement. Lipids are mostly carbon and hydrogen, with relatively little oxygen. They are nonpolar or mostly nonpolar, so they are hydrophobic. Lipids function in long-term energy storage, cell membranes, insulation, waterproofing, and hormones. Triglycerides contain glycerol and 3 fatty acids. Saturated fatty acids have no carbon-carbon double bonds and are straighter. They pack tightly and are often solid at room temperature. Unsaturated fatty acids have 1 or more carbon-carbon double bonds. These bends or kinks prevent tight packing and are often liquid at room temperature. Phospholipids have hydrophilic heads and hydrophobic tails. They form the cell membrane bilayer. Steroids have 4 fused carbon rings. Cholesterol is a steroid important in membrane fluidity and hormone production. Proteins are made of amino acids. Amino acids contain an amino group, a carboxyl group, a hydrogen, and a variable R group attached to a central carbon. The R group determines the amino acid's chemical properties. Proteins function as enzymes, transporters, receptors, antibodies, hormones, movement structures, and structural support. Protein function depends on folding. Folding depends on amino acid sequence and interactions among R groups. Picture proteins as precision tools. Their shape allows them to grip, cut, signal, support, transport, or catalyze. Nucleic acids are made of nucleotides. A nucleotide contains a sugar, phosphate group, and nitrogenous base. DNA stores hereditary information. RNA helps express genetic information and build proteins. DNA uses deoxyribose sugar and bases A, T, C, and G. RNA uses ribose sugar and bases A, U, C, and G. DNA is usually double-stranded. RNA is usually single-stranded but can fold into functional shapes. Nucleic acids function as information molecules. Compare the 4 groups: carbohydrates, sugars, and starches. Main roles: quick energy, storage, structure. Lipids, fats, oils, phospholipids, steroids. Main roles: long-term energy, membranes, insulation, hormones. Proteins, amino acid chains. Main roles: enzymes, structure, transport, signaling, defense, movement. Nucleic acids, DNA, and RNA. Main roles: genetic information, instructions for protein synthesis. If the exam asks for a biomolecule's function, identify its building block and shape. Sugar units point to carbohydrates. Fatty acids and hydrophobic behavior point to lipids. Amino acids and folded shape point to proteins. Nucleotides and genetic information point to nucleic acids. Track 08F. Enzymes in synthesis and degradation. Enzymes are biological catalysts. A catalyst speeds up a reaction without being used up. Enzymes lower activation energy. Picture a hill between reactants and products. Without an enzyme, the reaction must climb a high hill. With an enzyme, the hill is lower. The reaction happens faster. Enzymes do not change the overall energy difference between reactants and products. They change the pathway. Most enzymes are proteins, and their function depends on shape. The active site is where the substrate binds. The substrate is the molecule the enzyme acts on. The enzyme-substrate complex forms when the substrate binds to the enzyme. The enzyme may bring molecules close together, orient them correctly, strain bonds, or create a favorable chemical environment. Enzymes help synthesis. In synthesis, enzymes help join smaller molecules into larger molecules. Example: DNA polymerase builds DNA by adding nucleotides to a growing strand. Picture DNA polymerase as a moving assembly machine. It reads the template strand and adds complementary nucleotides. A pairs with T, C pairs with G. The enzyme's shape holds the DNA strand and incoming nucleotide in the correct position. Enzymes help degradation. In degradation, enzymes help break larger molecules into smaller molecules. Digestive enzymes break food biomolecules into absorbable units. Amylase breaks starch into smaller sugars. Proteases break proteins into peptides and amino acids. Lipases break lipids into glycerol and fatty acids. Nucleases break nucleic acids into nucleotides. Picture degradation enzymes as specialized scissors. Each enzyme cuts specific bonds in specific molecules. Enzyme specificity matters. Amylase does not break proteins. Protease does not break starch. Lipase does not break DNA. Specificity comes from active site shape and chemical fit. Environmental conditions affect enzymes. Temperature affects molecular motion and protein shape. Low temperature usually slows enzyme activity because molecules move more slowly. High temperature can denature enzymes by disrupting bonds that maintain shape. pH affects charge interactions in proteins. If pH changes too much, the active site shape or charge pattern can change. Each enzyme has an optimal temperature and pH. Inhibitors reduce enzyme activity. Competitive inhibitors bind to the active site and block the substrate. Non-competitive inhibitors bind somewhere else and change enzyme shape. Feedback inhibition occurs when the product of a pathway inhibits an earlier enzyme. This prevents waste and maintains balance. Enzymes are essential because cells need controlled reaction rates. Too slow, and life processes fail. Too fast or uncontrolled, and the cell loses regulation. Enzymes allow digestion, DNA replication, protein synthesis, cellular respiration, photosynthesis, signaling, repair, and recycling. For exam questions, use this reasoning chain. What is the substrate? What is the enzyme? What bond or molecule is being built or broken? Is the process synthesis or degradation? What happens if enzyme shape changes? If the enzyme is denatured, the active site changes. If the active site changes, the substrate may not bind. If the substrate cannot bind, the reaction slows or stops. Core rule: enzymes control life chemistry by matching shape to substrate and lowering activation energy. Track 0, 8, G, Rapid High Rigor Review. A through FA, elements necessary for life. The essential life elements are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Carbon forms 4 covalent bonds and builds molecular skeletons. Hydrogen and oxygen appear in water and organic molecules. Nitrogen appears in amino acids and nucleic acid bases. Phosphorus appears in ATP, DNA, RNA, and phospholipids. Sulfur appears in some amino acids and helps stabilize proteins. B. Water and carbon. Water is polar because oxygen pulls electrons more strongly than hydrogen. Polarity creates hydrogen bonding. Hydrogen bonding gives water cohesion, adhesion, high specific heat, high heat of vaporization, solvent ability, and ice that floats. These properties support transport, temperature regulation, chemical reactions, sweating, aquatic life, and membrane formation. Carbon is important because it forms 4 covalent bonds to itself, and creates chains, rings, branches, and complex shapes. Carbon allows molecular diversity. C. Molecular shape and biological function. Shape determines function. Enzyme active sites fit substrates. tRNA shape lets it connect codons to amino acids. DNA shape allows complementary base pairing and replication. Receptors bind specific signal molecules. Neurotransmitters activate receptors by shape and charge fit. Lipids organize membranes because hydrophilic and hydrophobic regions interact differently with water. Protein shape depends on amino acid sequence and folding. D. Synthesis and degradation. Synthesis builds larger molecules from smaller molecules. Dehydration synthesis removes water to form bonds. Degradation breaks larger molecules into smaller molecules. Hydrolysis uses water to break bonds. Cells synthesize DNA, RNA, proteins, membranes, glycogen, starch, and lipids. Cells degrade food, damaged molecules, storage molecules, and ATP. E. Biomolecule structures and functions. Carbohydrates are sugars and starches. They provide quick energy, energy storage, and structure. Lipids are hydrophobic molecules. They provide long-term energy storage, membranes, insulation, waterproofing, and hormones. Proteins are amino acid polymers. They act as enzymes, transporters, receptors, antibodies, signals, structural molecules, and movement molecules. Nucleic acids are nucleotide polymers. DNA stores genetic information. RNA helps express genetic information. F. Enzymes. Enzymes are biological catalysts. They lower activation energy. They are specific because active site shape matches substrate shape. Enzymes help synthesis by joining molecules. Enzymes help degradation by breaking molecules. DNA polymerase builds DNA. Amylase breaks starch. Protease breaks proteins. Lipase breaks lipids. Nuclease breaks nucleic acids. Temperature and pH affect enzyme shape and activity. Denaturation changes shape. Change shape changes function. Final lock-in. Life is chemistry. Elements build biomolecules. Water creates the environment. Carbon creates structural diversity. Shape creates function. Reactions build and break molecules. Enzymes control the speed and specificity of those reactions.